All right, if it's all right, I'll go ahead and get started. I'm Caitlin McGraw, and it's my express pleasure to introduce our very own Dr. Rane. Dr. Rane got her Ph.D. from the University of Akron, Ohio, and she did her postdoctoral fellowship here at the University of Louisville. She's currently an associate professor here in the Department of Medicine in the Division of Nephrology. And her research interests are centered on neutrophil biology in the context of acute lung injury, as well as diabetes-associated renal fibrosis mechanisms. Recently, in collaboration with Dr. Srivastava and Dr. Kai, her laboratory has begun investigating the mechanisms underlying effects of environmental toxins on renal injury and dysfunction. Her work has been funded by the NIH and the American Heart Association. So thank you all for joining us, and you may proceed.
Thank you so much, Caitlin, for the introduction. I would also like to thank Dr. Kai and Dr. Merchant for giving me this opportunity to present my work here today. So I'll first begin by talking about how diabetes regulates renal fibrosis and how eventually that led us to examining the role of environmental pollutants in modulating renal fibrosis. So the title of the talk today is Exploring the Potential Role of Nuclear Factor-Erythroid Derived Protein 2 or NFE2 in renal disease and injury. So as you all know, chronic kidney disease or CKD is increasing each year. If it is left untreated, it can lead to end-stage renal disease. And irrespective of the initial insult, fibrosis is the common pathogenic pathway that leads to organ dysfunction. And most importantly, the effective strategies to mitigate renal fibrosis are still limited. The number of people suffering from diabetes is increasing each year, and it's going to increase further in the next decade or so. The estimated deaths also caused by diabetes are increasing each year, and WHO has estimated it to be the 7th leading cause of death in 2016. Diabetes is also associated with a number of clinical complications, and which can be treated. Diabetes can be treated, and the complications can be avoided or delayed by just following a healthy lifestyle and taking medications. Why is it important to study diabetic nephropathy? It is one of the most common causes of end-stage renal disease, and it carries a huge economic burden. During diabetes, there is an increase in the synthesis of extracellular matrix proteins and a decrease in the degradation of ECM proteins. This results in net ECM protein accumulation, and this is mediated by TGF-beta and CTGF. Therefore, the goal of the laboratory is to understand the molecular and cellular mechanisms that regulate diabetes-induced renal fibrosis so that we can identify some molecular targets that can lead to generation of specific therapies to mitigate renal fibrosis. EMT, or epithelial to mesenchymal transition, is known to be a critical mediator of renal fibrosis. When the epithelial cells are injured, they transform into mesenchymal cells, and this leads to the progression of renal fibrosis. So during this EMT process, epithelial cells lose some of their proteins such as E-cadherin expression, whereas the mesenchymal phenotypic proteins such as N-cadherin, snail, and twist are increased. So what are adaptive and maladaptive repairs after injury? After the kidney undergoes injury, there is a cell loss due to necrosis or apoptosis, and surviving cells will proliferate, de-differentiate and proliferate, and they will restore a normal epithelium. This is an adaptive repair process. During the injury, if the injury is severe or repeated injury, and if the kidney epithelium is abnormal at baseline, then these cells do not proliferate as they get stuck in the cell cycle and they undergo G2M cell cycle arrest. These cells do not die by apoptosis, but they linger around and they induce activation of pathways such as junk MAP kinase pathway, which leads to the release of TGF-beta and CTGF, which then further activates myofibroblast proliferation. This leads to collagen production and accumulation of extracellular matrix proteins and fibrosis. How does TGF-beta promote tissue fibrosis? This can happen by a canonical pathway involving SMAD2, 3, and 4 complex, or by a non-canonical pathway which involves activation of MAP kinases, P38, JNK, and ERK MAP kinase. This leads to activation of transcription factors, which then modulate pro and anti-fibrotic proteins expressions. A number of approaches have been taken to abrogate TGF-beta mediated fibrosis. However, it is important to remember that along with being a pro-fibrotic factor, TGF-beta is also an anti-inflammatory factor. Therefore, complete blockade of TGF-beta mediated signaling can have serious consequences on the immune system, and therefore it is not a desirable target for intervention. Connective tissue growth factor is expressed in diabetic kidneys as well as in the urine of diabetic patients, and its expression is usually correlated with the severity of renal fibrosis. This led to a phase 1 clinical trial using an anti-CTGF antibody, and they found promising results as it was found to inhibit albuminuria. However, in phase 2 clinical trial, this trial was terminated by the sponsor due to suboptimal study design. Therefore, it is very critical to identify a target protein which lies between TGF-beta signaling and CTGF expression so that we can have targeted therapies to treat renal fibrosis. We were interested in the protein heat shock protein 27, as my laboratory and others have shown its role in modulating cell survival, regulating reactive oxygen species, which are an important mediator of diabetes-induced renal fibrosis. Moreover, HSP27 has been shown to regulate TGF-beta-induced oxidative stress and CTGF expression. Therefore, we wanted to understand the mechanisms by which HSP27 may modulate renal fibrosis. To do this, we performed a proteomics analysis to identify HSP27 binding proteins. This led us to identification of nuclear factor-erythroid derived protein 2, or NFE2, as an HSP27 binding protein. Now, what is NFE2? NFE2 is a transcription CNC cap and collar B-zip transcription factor that is known to regulate erythropoiesis, and it's also known to regulate beta-globin genes. It is also known to be present in the kidney, lung, and liver, but its role in these tissues is not clearly defined. NFE2 is a 45 kilodalton protein, and it's distinct from NERV2, which is NFE2 related factor 2. However, both P45 NFE2 and NERV2 heterodimerize with small MAF proteins. And once they heterodimerize, they induce gene transcription or they inhibit gene transcription. They are capable of either activating or inhibiting gene transcription. Also, a point to note is that the small MAF proteins can homodimerize and inhibit P45 NFE2 transcription. So they compete for the binding site of P45. Since NFE2 was found to be an HSP27 binding protein, we first wanted to examine its expression in human renal proximal tubular cells that were treated with TGF-beta, or in the diabetic kidneys, type 1 and type 2 diabetic kidney homogenates. As you can see, TGF-beta decreased NFE2 expression in the HK11 cells. Similarly, we saw a decrease in NFE2 expression in type 1 diabetic kidneys as well as type 2 diabetic kidneys, with a concurrent increase in fibronectin expression. HSP27 has been shown to bind ubiquitinated client proteins and target them for degradation. Moreover, phosphorylated HSP27 has been shown to target the client's proteins to proteasomal degradation. This led us to hypothesize that TGF-beta induced HSP27 phosphorylation promotes NFE2 degradation and CTGF expression. So if you see here, if NFE2 and MAFK bind to the promoter sequence of CTGF, they may possibly inhibit transcription of CTGF. And I say this because NFE2 binding sites are present in the CTGF promoter. However, it's not proven whether it can bind and repress CTGF expression. Similarly, the two MAF proteins can homodimerize, and they have been shown to induce CTGF expression. So in the absence of NFE2, when it's degraded, it's possible that the two small MAFK proteins can homodimerize and induce CTGF expression. To examine the role of the proteasome in degrading NFE2 expression, before that, we first wanted to see if NFE2 is repressed at the transcript level, or if it's regulated by degradation of the proteasome. So we first performed a PCR, RT-PCR, to examine the expression of NFE2 transcript in control and TGF-beta treated cells, and these results demonstrated that the transcript was formed, but the protein was possibly degraded at the proteasome. So the cells were treated with an increasing concentration of Mg132, which is a proteasome inhibitor. And in a dose-dependent manner, you can see that NFE2 expression is preserved. In the absence of Mg132, NFE2 degradation is seen, and in the presence of increasing concentration of the proteasome inhibitor, you see that NFE2 expression is preserved. Interestingly, as NFE2 expression increased, we saw a decrease in CTGF expression and a decrease in fibronectin expression. Also, we have shown similar studies were done in animals that were treated with Mg132 for 3 months. Type 1 diabetic OVE26 mice were treated with Mg132 for 3 months, and the kidney homogenates were immunoblotted for CTGF and NFE2. We were able to demonstrate the same results in the kidney homogenates, where in the presence of Mg132, NFE2 expression was preserved and CTGF expression was decreased. Next, we wanted to examine if NFE2 degradation was mediated via its binding to phosphorylated HSP27. So we first treated HK11 cells with TGF-beta, and we immunoblotted it with phospho-HSP27 antibody to demonstrate that indeed TGF-beta stimulated HSP27 phosphorylation in these cells. We then performed an anti-NFE2 immunoprecipitate in these samples, along with an isotype control antibody immunoprecipitation, followed by immunoblot with phospho-HSP27 antibody. As you can see, HSP27 and NFE2 associated in control cells, and this association was significantly increased in the presence of TGF-beta, suggesting possible role of HSP27 in targeting NFE2 to the proteasome. Next, we wanted to examine the effects of overexpressing NFE2 on profibrotic signaling in HK11 cells. The HK11 cells were transfected with either the vector alone or with NFE2 constructs for 24 hours, and then they were treated with TGF-beta for additional 24 hours. As you can see, in the presence of TGF-beta, you see an increase in fibronectin expression, which was abrogated in the presence of overexpressed NFE2. Similarly, we saw a decrease in CTGF expression and significant decrease in Pi1 expression in these cells. These results suggest that overexpression of NFE2 was able to mitigate TGF-beta-induced profibrotic signaling. We next silenced NFE2 expression to see the effects of silencing NFE2 on TGF-beta-induced CTGF expression in these cells. In the presence of control siRNA and TGF-beta, we saw an induction of CTGF expression, which was exacerbated when NFE2 was expression was downregulated by siRNA. To summarize this part of the talk, HSP27 is phosphorylated in TGF-beta treated cells, which causes it to associate with NFE2 and possibly degrade it at the proteasome. In the presence of MG132, an inhibitor of the proteasome, we were able to show that NFE2 expression was preserved. Overexpression of NFE2 blocked Pi1 fibronectin as well as CTGF expression in TGF-beta treated cells, whereas silencing NFE2 expression resulted in exacerbation of CTGF expression in TGF-beta treated cells. We next wanted to know what were the mechanisms by which NFE2 degradation occurred in these cells. We hypothesized that the non-canonical TGF-beta signaling pathways controlled NFE2 expression in these cells. We first examined the effects of TGF-beta treatment on ERK and P38 phosphorylation. And as you can see, TGF-beta significantly induced ERK and P38 phosphorylation in these cells. We then we then inhibited just P38 in HK11 cells prior to stimulating them with TGF-beta to determine the effects of P38 blockade on NFE2 expression. What we saw in these studies was that blockade of P38 partially preserved NFE2 expression. Therefore, since both P38 and ERK were activated, we treated the cells with P38 and ERK inhibitor, MEK-ERK inhibitor, followed by stimulation with TGF-beta. Blockade of both ERK and P38 pathways resulted in loss of TGF-beta induced HSP27 phosphorylation, and it completely preserved NFE2 expression, suggesting both pathways contribute to NFE2 degradation. We next examine the effects of blocking ERK and P38 MAP kinase pathways on TGF-beta stimulated fibronectin and CTGF expression. TGF-beta stimulated fibronectin and CTGF expression was abrogated by blockade of both the pathways, suggesting that ERK and P38 pathways modulate expression of NFE2, fibronectin, and CTGF. Next, we examined the role of JNK pathway in modulating NFE2 and CTGF expression. JNK has previously been shown to induce CTGF expression. Therefore, we were not surprised that blockade of the CTGF, blockade of junk MAP kinase pathway inhibited TGF-beta induced CTGF expression. However, what was surprising to us was the fact that blockade of junk had no effect on NFE2 expression. This suggested to us that possibly junk MAP kinase pathway was activated downstream of NFE2 degradation. we examine the effects of blocking the proteasomal activity on TGF-beta induced junk activation. So TGF-beta mediated junk activation was significantly decreased, inhibited in the presence of MG132, which is the proteasome inhibitor. Similar results were obtained in OVE26 mice that were treated with MG132. So the junk MAP kinase pathway was activated in OVE26 mice, which was blocked by treatment with MG132. These results suggest that junk MAP kinase is activated downstream of proteasome activity. Next, we wanted to see the effects of overexpressing NFE2 on TGF-induced junk phosphorylation. So we transfected these HK11 cells with PU's vector or NFE2 constructs, and then followed it up by treating the cells with TGF-beta. TGF-beta induced junk phosphorylation was significantly inhibited in the presence of overexpressed NFE2. Having shown the fact that NFE2 overexpression, underexpression regulated CTGF and ERK and P38 MAP kinase pathways promoted its degradation, we next wanted to examine the effect of overexpressing NFE2 in the kidneys of diabetic mice. So initially we generated a purified NFE2 protein in the laboratory, and we transduced these proteins in FVB mouse kidneys and demonstrated the presence of NFE2 in these mice. In the future, we plan to transduce these proteins into the OVE26 mice as well as in DBDB mice to determine if we could possibly use NFE2 as a therapy for management of renal fibrosis in diabetic mice. To summarize this data, TGF-beta induced P38 and ERK phosphorylation, which led to HSP27 phosphorylation. Phosphorylated HSP27 associated with NFE2, and this possibly led to its degradation at the proteasome. Blockade of the proteasome resulted in blockade of junk MAP kinase pathway and blocked the activation of CTGF. Overexpressed NFE2 blocked junk activation, and we have previously shown that NFE2 overexpression also blocked CTGF expression. To conclude this part of the talk, TGF-beta induced P38 and ERK MAP kinase activation promotes association of NFE2 and phosphorylated HSP27, resulting in NFE2 degradation at the proteasome, which ultimately leads to junk activation and CTGF expression. We are also examining currently the role of epigenetic modifications in the regulation of NFE2 expression and renal fibrosis. We are also examining the role of phosphomimicking and phospho-dead mutants of HSP27 in regulation of NFE2 expression and renal fibrosis. We are also identifying role of renal AKT substrates in modulating different stages of renal fibrosis. Moreover, given the fact that cigarette smoking can cause diabetes, we are also examining the effects of environmental pollutants such as cadmium and acrolein, both constituents of cigarette smoke, on renal fibrosis. In collaboration with Dr. Lu Kai and Zhao Li, we are examining the effects of whole life cadmium exposure on mice that were treated with low fat and high fat diet and determine its effects on renal fibrosis. So initially, we examined the effects of high-fat feeding and cadmium exposure on albuminuria and the ratio of kidney weight by tibia length in mice that were at the 24-week male mice. I forgot to say here how these mice were treated. So the male and female mice were first kept on a water regimen, tap water, which was control water, and water that was treated with 0.5 ppm cadmium or 5 ppm cadmium 1 week prior to breeding. Along with, they were continued on the same regimen during pregnancy and weaning. Once the offsprings were weaned, they were then put on low-fat diet or normal diet and high-fat diet. And then the animals, the mice were sacrificed at 10 weeks or at 24 weeks. What we see here is that we see urine albumin is increased in mice that were treated with high-fat diet alone, and high-fat diet plus high cadmium mice significantly exacerbated this response compared to high-fat diet alone, and this was significantly decreased by low cadmium exposure on mice that were treated on high-fat diet. Similar results were obtained for kidney weight ratio to tibia length, where high-fat diet induced this KW by TL, and which was enhanced further by high cadmium exposure and was significantly decreased by low cadmium exposure in the high-fat diet treated mice. Similarly, high-fat diet feeding and cadmium exposure also enhanced fibronectin and CTGF expression significantly higher than high-fat diet alone, and low-fat diet significantly decreased this response. We are currently examining the other parameters of renal fibrosis in these animals, and hopefully in the next talk, I will present more data on these studies. On a separate project that I collaborated with Dr. Srivastava and Dr. Conklin, these mice were treated with Acrolein or charcoal filtered air for 12 weeks. And they were sacrificed after 12 weeks, and the kidney homogenates and tissue sections were collected and were subjected to immunoblot analysis for NFE2 and CTGF. Similar to my studies in diabetic animals, we saw that 1 part per million acrolein exposure decreased NFE2 expression and induced CTGF expression. Moreover, acrolein treated mice had a significant increase in serous red staining as compared to mice that were treated with air. H&E staining also demonstrated some inflammation in acrolein-treated mice, some interstitial proliferation and interstitial matrix deposition, suggestive of renal activation of renal fibrotic pathways. We are currently waiting for results from a proteomic study that Dr. Mike Merchant performed on the control and air and acrolein treated mice kidneys to see differential protein expression after acrolein treatment. We also are waiting on RNA-seq analysis data on these samples that were made possible due to a medium voucher grant that was supported by the P30 NIEHS grant awarded to Dr. States. Before I finish, I would like to acknowledge people in the laboratory, Shunyin Jin and Jia Li, who did most of the work. Some of the work was also done by Rachel Wu. I would like to thank Dr. Chai, Yi Tan, and Evelyn for their support, Dr. Chai particularly for all the help with the projects involving cadmium as well as diabetes. He is very instrumental in most of the work that was presented here today. His input was important. I would also like to thank Michelle Barati and Dr. Merchant for their constant support. And I would definitely like to thank Dr. Srivatsava and Dr. Conklin for providing samples of air and acrolein treated mice. Thank you for your attention. I'd be happy to answer some questions.
This is Krishna Ajala here. Nice talk. Just have a quick question. Did you like checked with any NRF2 activators in this model to see if would enhancing the NRF2 activation would benefit?
That's a good question. I haven't examined NRF2, but this is something I plan to examine with Dr. Michelle Barati, who does work on NRF2 signaling in the kidney. So NRF2, NFE2 was first identified, and then NRF2 is an NFE2 related factor 2. However, they both regulate a number of common targets, such as antioxidant genes like glutathione, synthetase, and catalase. But NFE2 regulates a wide variety of gene targets such as caspase-12, RAB27, some integrins. So the range of proteins that are regulated by NFE2 are very different from NRF2. So it'll be very interesting to see what we get with NRF2. My guess would be they would be all detoxifying enzymes like HO1, NQO1, and targets that are involved in the detoxification process. Do you have any other?
Yeah, I mean, we have NRF2 knockouts and we work extensively on NRF2 targeted pathways in the variety of diseases. So, I mean, maybe we can chat more, so.
Yeah, absolutely.
I mean, yeah, we work with the microbial metabolites, and then those are the ones which are, some of them are activates, not to the extent where it becomes toxic, but it has a moderate activator where it can only give the benefits of the antioxidative pathways. So maybe we can chart, definitely it's, I mean, I never worked with the kidney, but I worked with all the in the gut. So
yeah, definitely. Michelle would be very happy to hear this because she's very passionate about NRF2 signaling.
Yeah, we
definitely
have all
we
the
should
tools
chat.
we have all the tools to check. Okay, so that would be awesome because we found new microbial metabolites that can really activate the NRF2 pathways to. Mainly, we focused on gut barrier. I mean, I bet it will have a role in the kidney.
Okay.
So, yeah.
Cool. That sounds good.
Yeah, we can talk maybe.
Definitely.
Yeah. Thanks.
Thank you.
I'll ask a question.
Okay.
Great. Hi. So I'm seeing something very similar in the cadmium model in the liver, in which the higher dose of cadmium is exacerbating the diet-induced response, and that the lower dose of cadmium is actually making it better.
Yeah.
I was wondering if you had any thoughts as to why that's happening.
That's a great question. There is all these pathways that are being activated, like the autophagic pathway. We are trying to now look at different mechanisms of cell rescue, like, if autophagy is induced in these cells as a mechanism to protect them from apoptosis and what have you. So there are different pathways that we have outlined that we need to examine, but we can chat later about the specifics.
Okay, sounds good. Thank you.
Hey, Madhavi. Yeah, this is Lukai. And
Yeah.
Because Jimmy asked that question because we are working on the liver, and the model is same model, and but we are working on the different organ. And Jimmy worked on the liver. We found a significant like a protection in the low dose ketamine treatment group, and we try to work on the like antioxidant pathway, measure some like antioxidant level, especially methylcyanine. And I think that in the kidney, we need to think about that too, not just autophagy. And the low dose ketamine probably turn the methylcyanine and levels, so that somehow probably play some role. Yeah.
So in the preliminary studies that I did, I had found an increase in NFE2 expression in the low dose. So I was kind of excited.
So that is
but we did not have enough ends on that to kind of say it. But since you are asking, I'm telling you that Gia had found that initially, when I sent the, we sent you some data, but we need to confirm that with more ends.
Sure. Yeah. Yeah.
But
this,
so
this is a link. So, liver, kidney, and there's a low dose, high dose difference. Yeah. Very good.
Yeah.
Hey, Madhavi.
Hi.
Hi. Thanks for a really interesting presentation. I didn't know about the results of the chronic inhalation of 1 ppm acrolein produced such increase in serious red staining in the kidney. So this is really interesting. In terms of, I'm just kind of wondering about the timing of events and what happens first. So when you take samples at just like one time point at 12 weeks and you see one thing go up and one thing go down, the question is really what happens first, and how do you speculate that it unravels, and how does acrolein, because many times people put these alpha beta unsaturated aldehydes on cells, it stimulates a very strong NRF2 response, upregulates the antioxidant enzymes, and GSTs, for example, go up. But in vivo, it looks like it's much more complicated. So I'm wondering how you think the timing occurs with this process.
I mean, the initial antioxidant response is an adaptive response, right? So you would see all these things going up, NRF2, but it would be important to see what happens to NRF2 as you go with, with increased timing. You might actually see a decrease in NRF2 signaling.
But why do you think that is? What do you think happens?
So I think the initial increase is because acrolein causes depletion of glutathione, and so it causes oxidative stress, and that causes NRF2 to be upregulated. And but as the acrolein exposure continues, this becomes more chronic, and then it's not strong enough to combat that response, and so you lose NRF2 activation probably with time. So I think it's it's just because there's so much oxidative stress, and then you see NRF2 going up to combat this oxidative stress, but it's not strong enough to continue to mitigate it.
So
does that make sense?
Well, I don't understand why it would do that necessarily. And do you have any evidence that everything else is dependent on that? So does NRF2 signaling have to go down in order to stimulate fibrosis, or are they completely unrelated events? That's one question. And then did you measure any of the antioxidant enzymes that are under NRF2 control in the kidney that show that they're actually being downregulated?
Okay, so first of all, I think that the data I showed was NFE2 and not NRF2.
Right.
So that's always confusing. I don't blame you, but NFE2 is different from NRF2, and I did not measure NRF2. I measured NFE2, and NFE2 goes down, and the results that we've got, I mean, I just got something from Mike where he did a proteomic analysis on air and acrolein treated kidneys. And basically, I haven't sat and analyzed the data properly yet. But what his initial the figures show is that in the presence of acrolein, you see decrease in glutathione synthetase, you see a decrease in catalase. With decrease in NFE2. So less NFE2, these are targets of NFE2, they also downregulated in these 1 ppm samples, acrolein treated samples.
Yeah, no, I think that's a, so is that necessary to increase fibrosis? Is it a mechanism by which fibrosis is enhanced, or are they separate events?
This is a good question. I mean, it could be true. I mean, for the longest time, I kept thinking it could be true, unrelated, that you overexpress NFE2 and you decrease all the profibrotic signaling. But when you silence NFE2 also, you see that, you exacerbate the response. Now, the way to test it is I've generated NFE2 recombinant protein. So we are ready to try and deliver it to diabetic mice and see if it'll mitigate its fibrotic signaling in the kidney. I mean, that is the only way to know if it's doing something. And if these two are, so for example, here's the thing, NFE2 will replenish the glutathione pool by inducing glutathione synthetase, which will then lead to more glutathione that will reduce oxidative stress signaling, and that would alleviate fibrotic signaling downstream. So it's possibly connected. And if it does not alleviate fibrosis, then you can just say that this is true unrelated, because we are going to just reduce oxidative stress, but fibrosis continues to happen.
Right. I think that's what I was kind of getting at about the timing too.
Yes.
Like if we did some at 4 weeks or 8 weeks, we
absolutely. So that's a grant waiting to happen.
Yeah,
we should talk more.
Great. Thank you.
Thank you.
That's a very neat result. Because
thank you for all the samples. Thank you for all the samples, Dr.
No problem. We're glad you could look at them. I mean, those mice, when they come out of that, they look phenotypically normal. So we don't see like a big change in the animals in terms of their growth or general health. So it's not like they're really sick, but you're clearly observing something that could be very important to pathogenesis with chronic exposure to VOCs like Acrolein.
Right. I was surprised too when I saw that. But now when I see the proteomic data, I mean, a lot of it is not statistically significant. We got, I think, 65 statistically significant changes. But Mike said, if Mike is there, he can chime in. But he told me if we increase the N, you can get significance. But proteomic analysis is just like a guide. It'll lead you to proteins which you validate by doing immunoblots or PCR or what have you. So it's just a guide to let you see which pathways are modulated. And really, I found some very interesting, like FGF1 I saw was decreased, decrease in acrolein treated mice, and FGF1 is known to alleviate renal fibrosis. So things like that. There are very many interesting proteins which have shown up, which showed up in this proteomic analysis. But I have really not had the time to, because I just got it from Mike, so I have to spend time with him to analyze the data more.
Yeah, so this was a pilot study. So it was, looking at the medium high abundant proteins. And when Madhavi was saying that they weren't statistically significant, they were 0.06. It was 0.06. And so, we had to take it up to an N of 5. This was just an N of 3 versus 3. And it's pulling up some very interesting proteins. One or two she's developing that look very interesting, but the whole glutathione angle was clearly there.
Yeah, that's great. So just for point of clarification, these were all male mice? Madhavi? We didn't,
Yes, male mice.
Okay,
Yeah.
do you think there's any sex dependence of these kind of responses, or?
Could be. I haven't tested it.
Okay. I think we may have samples from females as well. We might look at, so.
That would be cool.
All right, thanks so much.
Thank you. Okay. I guess there are no more questions then.
Dr. Kong, you there?
Yes, I'm here.
Yeah.
If there are no more questions, then I guess we'll close this seminar.
Thank you. Thank you.
Yeah, hope to see you back here next week.
Thank you.
Thank you so much.
We have multi-range meeting at 12.
So is it on the same chat, or we have to go sign out and sign back in?
Separate one. You
should have a separate calendar invitation.
Okay.
See you.
Thank you.
Thank you.
Nice talk, Madhavi. Good job.